Hi, welcome to our next session. Our next speakers will be both Michelle Gill and Avantika Lal. They are both senior scientists at the Deep Learning Proteomics group at NVIDIA. Today, they will be talking about real-time GPU accelerated analysis and visualization in the life sciences. Thank you guys for being here. I'm looking forward to your talk.
Thank you, and thank you, Angela. And I'd like to obviously thank the organizers for the opportunity for us to share our work today. So today we're going to discuss the role that bioinformatics plays in the drug discovery process and how GPU accelerated methods can help enable those roles and further advance them. We're going to demonstrate this with two examples. The first from single-cell RNA analysis, and then one pertaining to virtual screening. And both of these examples relate to the ongoing COVID-19 pandemic. So cells contain genetic material known as DNA. And an organism's total DNA is known as its genome. And the study of the genome is known as genomics, as you can see at the top. The contents of an organism's genome control many of the traits. In the case of humans, they control many of the traits that make us human, that make us unique from each other. And the human genome itself contains thousands of genes, approximately 20,000, which are transcribed into RNA. This RNA is then translated into proteins. And proteins make up the various aspects of a cell which catalyze chemical reactions. The products of these chemical reactions from proteins are often known as metabolites. So the study, then to back this up, the study of RNA on a broad scale is known as transcriptomics. The study of enzymes and all of the gene products within an organism is known as proteomics. And the study of the metabolites produced by chemical reactions is known as metabolomics. So today we're going to cover transcriptomics, as you can see in the middle, and we will also cover cheminformatics, which is very similar to metabolomics, but is really a broader family of techniques. And it's more associated with the data science aspect of studying small molecules. So things associated with the search and the retrieval of chemical compounds. So the drug discovery process is long and expensive, and I certainly don't have to tell many of you that who have experience with science or who are eagerly following the search for a vaccine for the ongoing pandemic. What you see here is what the discovery process looks like for small molecule drugs. And conservative estimates have it at around $2 billion and 10 years of research. So again, a very long journey to discover a drug for the treatment of a disease. And the computational and informatics methods that we just discussed are in fact an integral part of this process. You can see genomics and transcriptomics associated throughout the process, proteomics, various simulation, molecular dynamics, and cheminformatics as well. So the growth of these techniques, these omics techniques, as we call them, and informatics techniques, is accelerating the pace of scientific discovery. And it's also, in a way, trying to accelerate the drug discovery process. Estimates are right now, due in part to big data techniques and to also advances in equipment, scientific equipment as well. Estimates are that the global scientific output doubles every 9 years. So clearly so again, just to reinforce, this is effectively the growth of these techniques can help bring a drug to market, but it also creates a challenge. We need high-powered tools and highly parallelized tools to help us sort through and make sense of all this data. And that's really where data science in general comes in. It provides a toolkit that we can use to make sense of our results and to find the needles in the haystack when necessary. However, the growing need in science to increase the throughput and the acceleration of these tools is creating a need for more computationally efficient hardware and software. And GPUs are an ideal platform for this need because they have because of their highly parallelized ability to perform computations. On the software side of things, an ideal solution is what we call our toolkit of GPU-accelerated data science packages. And this is called Rapids. So for each of these packages, there is a GPU-accelerated library that is very analogous to what exists in the Python world that is popular for performing data science and machine learning, graph analytics, and visualization on more traditional methods. And so there's something parallel that allows on GPU data science to be performed for each of these tasks. And now I'll turn it over to Avantika.
Thanks, Michelle. So our first example concerns single-cell transcriptomics. And single-cell transcriptomics is a method by which we can isolate single individual cells from a biological sample, such as a part of the human body. And having isolated single cells, we can measure the RNA content of every cell. By measuring the RNA content, that is, in effect, a measurement of the functional activity of every single gene. So we can measure the activity of every single gene in the genome, and we can construct a matrix like the one shown here. And in this matrix, we store the activity of every gene in the human genome for every single cell in our biological sample. What do we do with such a matrix? Well, we can cluster the rows of this matrix, and that allows us to identify clusters of cells or groups of cells that have very similar gene activity to one another. And these clusters of cells that we can identify correspond to different biological types of cells, which perform specific functions inside the human body. Next slide. So here's a more detailed workflow of the analysis that we perform on a single-cell transcriptomic count matrix. We have this matrix which shows the activity of different genes across different cells in our sample. And we can start by normalizing and correcting this matrix. And then we typically perform principal component analysis, or PCA, which reduces the dimensionality of the matrix. Then we can use any of a variety of different clustering algorithms to cluster the rows of this matrix and identify groups of similar cells. Next, we can visualize the clusters that we discovered in a two-dimensional embedding using UMAP or TSNE. And finally, we'd want to perform a differential gene activity analysis, which means that we would want to compare the different clusters to understand what makes a given cluster different from all of the other types of cells. Next slide. Okay, so identifying different types of cells in the body with single-cell transcriptomics can be very important for drug discovery. And one reason why this is so is because many diseases affect specific types of cells in the human body, or have different effects on different types of cells. And similarly, many drugs or other therapeutics affect specific types of cells in the human body. So it becomes very important for us to know what the different types of cells in the body are and how these different types of cells are affected differently by various perturbations, so that we can develop more efficient and targeted treatments for diseases. And COVID-19 is actually a really good example of this. So if you look on the top right, you see a clustering plot. And this, in the beginning of the pandemic, scientists analyzed a lot of single-cell transcriptomic data from the human lung. And this plot on the top right shows you the clusters of human lung cells that were discovered from such an experiment, showing the different types of cells that form the human lung. And below, you see box plots representing the expression of the ACE2 gene in each of these different cell types. And the SARS-CoV-2 virus that causes COVID-19 actually requires the ACE2 gene, or the product of the ACE2 gene, in order to infect a human cell. So this tells you that there are different groups of cells in the human lung which carry out different functions, and that some of these groups are more susceptible to SARS-CoV-2 infection than others. Next slide. Okay, so at NVIDIA, we have developed a pipeline to analyze single-cell transcriptomic data using Rapids, therefore analyze this data on the GPU. Here we see that in the table, we see some of the different steps of the analysis that I outlined earlier. And next to them, we see the runtime of these steps in seconds on a multi-core CPU instance. And next, we see the runtime of all of these steps in seconds on a single GPU. Both of these columns is the runtime of all of these steps in seconds on 2 different GPUs. And in brackets, you see the acceleration. So that tells you that many of these steps can run hundreds of times faster using a single GPU. For example, a clustering of cells using the Louvain algorithm, which takes 10 minutes on a CPU instance, can be run in under 3 seconds on a GPU. Next slide. So let's not start the video yet. So here I'm going to give you a demonstration of how fast GPU analysis of single cells can be. Here we are going to visualize the clustering of 70,000 human lung cells and analyze this data using rapids. Let's start the video. So over here you can see the clustering of 70,000 cells. And below, you can see a plot representing the expression of the ACE2 gene, or the activity of the ACE2 gene in each cluster. And this shows you that there are some clusters of cells in this dataset which have higher activity of ACE2, and which therefore may be more susceptible to SARS-CoV-2 infection. We can select any particular cluster that we are interested in and just click a button. And within seconds, when we click this button, we will select this group of cells and perform clustering again on the selected cells. And this allows us to instantaneously identify that there are different smaller subpopulations within this type of cell. And if you look at the plot below, that these smaller subpopulations also have different activity of the ACE2 gene, indicating that there are smaller groups of cells which might also differ in their susceptibility. So I hope that this example shows you how GPU acceleration can make biological data analysis much faster, easier, and more interactive. And I will hand it back to Michelle to show you a second example.
Thanks, Avantika. So let's move on to Cheminformatics. So Cheminformatics is associated with both computational, so in silico, and experimental aspects of drug discovery. One common computational use that we're going to be talking about today is virtual screening. And in the process of virtual screening, large libraries of compounds are docked in silico, again, computationally, to a 3-dimensional structure of a protein target, the purpose of which is to determine which compounds are predicted to bind. Successful predictions, usually as determined by some sort of computational scoring method, are known as hits. So those hits are then prioritized for experimental validation. So compounds can be synthesized and tested in an experimental assay, and the process can be repeated many times. Cheminformatics is also used, however, for the evaluation of in vitro and in vivo experimental results. So these very same assays, which are conducted on the virtual screening hits, could then utilize Cheminformatics in the analysis stage to help, again, use data science techniques to pull out the common threads and help us understand the insight that's present in large amounts of data. One challenge with small molecules and with chemicals is how do we featurize chemicals in a way that is amenable to modeling, to machine learning and deep learning. And so that's what we'll talk about next. And we're going to be focused on the specific type of featurization that we'll be using in the demo. However, please know that there are many types of methods that can be used to featurize molecules. So what we're going to talk about today is known as a Morgan fingerprint. It is a very commonly used and very popular method for converting a compound, like you can see here on the left, into something that is a numerical form that we can use for machine learning. So with Morgan fingerprints, we take increasing numbers of atoms. So you can see, for example, on the top row, we're taking effectively one atom at a time that's from this molecule that we want to featurize. And then progressively, we take larger and larger numbers of atoms from the molecule. And for each of these atoms, we use a variety of features, such as the element, so oxygen or nitrogen, carbon, and we also use other features such as the connectivity of the atom, et cetera, et cetera. So for increasing numbers of atoms. And so each of these sets of features is hashed to create an identifier like you can see on the right. So this is just a numerical array of identifiers that exist for each of these sets of features. These identifiers themselves are then hashed once again to produce a fixed length binary array. And this binary array that's of universal size, regardless of our input chemical, is what we use for machine learning. And in some cases, this binary array is used directly. In some cases, it may be featurized further. But hopefully this gives you a flavor of how this process works with machine learning. And one other point to note, there can be collisions depending upon how big this bit array is. So you can see two examples of collisions happening here. So let's take a look at the workflow that we'll be covering today in the demo for the Cheminformatics project. So we start with a compound library, and we're using a publicly available library known as Chemble. There are approximately 1.9 million compounds in Chemble. And the data processing, as we just discussed, is used to create Morgan fingerprints. And this uses an open source library called Python. And then after the fingerprints are created, we then move the data onto the GPU. And so the steps right now that our demo undertakes, the first is dimensionality reduction with PCA, just like the single cell RNA demo. We then perform clustering with k-means, and then an embedding is created that's used for visualization with UMAP. This feature set right now is small. This is relatively new work, but we certainly anticipate growing the capabilities of the Cheminformatics workflow as it becomes more popular and as we get feedback from users. So here's the data that we'll be using for the demonstration today. So this is a very recent paper, and the author of this paper performed a virtual screen like we just described using Chemble as the database source, and identified about 27 hits. Again, this is with in silico modeling for compounds that were found to bind to a specific protein from SARS-CoV. And you can see several of these hits in the table on the left. There's actually about 27 of them. So this is just a subset of the chemical structures and their Chemble IDs, and then what their known targets previous to this instance are. On the right, you can see a three-dimensional structure. This is the protein that was used for docking from SARS-CoV. It's a specific protease that is indeed a known potential drug target for COVID-19. And you can see an example of one of the Chemble compounds docked into this portion of the protein receptor. So to begin, so just before I start the demo, this example is showing 10,000 molecules. However, I'm able to scale this demo up to around 500,000 molecules on a single GPU, and there are minimal delays in the clustering steps. I'm showing a smaller number really for the purpose of visualization. With large number of molecules, it becomes a little bit difficult sometimes to see molecules that are highlighted of interest. So again, this is our clustering step, and I will insert on the right, as you can see, the Chemble IDs that were identified as hits. And now we are zooming in. And just to point out, this clustering is all being done live on the GPU instantly. And you can see the hits being highlighted with the large diamonds. And I can zoom in again. And you can see, I'm just going to pause the demo for a moment. You can see the hits again shown in large and their various clusters. Upon mouse over, you can see each of the Chemble IDs that correspond to the specific molecule which is shown up at that position within the clusters. Down below, you can see part of the table, which we'll scroll through in a minute, that contains the molecular structures, the IDs, and there are links to the Chemble information, as well as a variety of other data that can be added optionally. So we can then zoom in on on these, and we can select, say that there, for example, maybe there's 3 that are of interest to us specifically because they happen to have very similar chemical structures. Again, we're reclustering in real time, and we can scroll down and see each of the compounds that we've selected. And let's say, for example, we know of another compound, either one that we saw above in the field of view, or one that we happen to know that it's similar. We can add it and then scroll down, and you can see that this compound also has a structure that's relatively similar. So the potential use case here, again, is examining the results from virtual screens. Perhaps scientifically, one wants to explore those results further. So, and that concludes the Cheminformatics demo, and as well as our talk. There are a variety of individuals that we'd like to thank who made this possible. Pat Walters from Relay Therapeutics, who provided some feedback on a prototype, as well as a variety of individuals from NVIDIA. You can see the GitHub repos, and again, both of these start with http://github.com. But the top is the rapid single-cell RNA demo, and the bottom is the Cheminformatics demo. And you can also find us on Twitter. These are our handles on the right. Thanks, and we would be happy to take questions.
Thank you guys so much. I think for me, it really just stands out that, like, when we talk about precision medicine, it's like I can see a time when, when you show your results that we will actually be able to, do the patient's analysis when they're sitting in the office. I mean, are there other types of analysis that you're focusing on that would help a patient day to day?
Avantika, I can let you answer that first if you want.
Yeah, I think, so Michelle talked about Rapids, and Rapids is basically a set of Python libraries that can be used for all sorts of data science. It carries out the same functions as common Python libraries that people use every day for data science, like Pandas or scikit-learn. So you can analyze any kind of tabular data, perform machine learning, integrate with deep learning frameworks, visualize your data in different ways. So it's a very versatile functionality that can be applied to analyze any kind of patient data.
Yeah, go ahead. Sorry. I was basically just going to follow up on that. I mean, because that's based on what you're saying, it's actually not that this is useful for application of current algorithms, but this actually could help develop new algorithms. Because a lot of times when we're developing, we have to do things 1,000 times, and then we know what our new method is.
Yeah, I mean, maybe I can take this one. Absolutely, I think that's the beauty of Rapids, is that it's designed to be very familiar, but also to allow rapid experimentation. Maybe, hopefully not irritation, but. In machine learning, many times we have to tune parameters, and even that you can tune it on a much larger portion of your data, and you can do it real time. So you're able to find, improve your results, improve outcomes. It really is a foundational toolkit for all of machine learning, and certainly there are very strong applications to diagnostics, to personalized medicine, as you said.
Did you have problems mapping the docking algorithm to the GPU? Are all algorithms amenable to this?
So just for clarification, so the docking was done as part of that publication. We did not repeat the docking on the GPU. There are various software packages, not necessarily a part of Rapids, that do that type of work, both on the CPU and some on the GPU. But we were merely, we took the results and we wanted to examine all of these results in the context of the database that was used for docking. So our focus was really exploration of the data in the context of clustering and visualization.
We have a question about the demographics. How are the demographics considered in the population sampling?
Avantika, I'll let you take that.
Right. So thinking of the single-cell RNA-seq dataset, yes, if we want to, so here we are just clustering the data and identifying groups of cells. But if you have multiple individuals, then demographics is one of the things that you need to take into account in such studies. And the way we do that is sometimes that we include demographic factors as covariates in a machine learning model if we are trying to predict some outcome from the single cell data. We can also include it as part of the normalization and correction steps that I mentioned in the beginning of our analysis workflow, where we can take groups of cells that come from different patients and try to correct them to account for interpatient variability that could be due to demographic factors.
Thank you guys so much. I think we've run out of time. I hope I get to see you guys again soon.